This does work, doesn't it? And welcome, everyone, and everybody who's joining us online. Welcome to the Headwaters lecture. This is actually the second in this event series, so it's a relatively new thing that we started this year. Basically, the purpose of the Headwaters lectures is to bring internationally known field-shaping researchers here to campus to be able to interact with what I call the water community of faculty and early career scientists and students. And the name might be obvious, Headwaters Lecture. So we're here at in geographic space in Minnesota at the headwaters of three major river systems. Kind of the purpose of this seminar series is to have a discussion at the headwaters in the space of ideas in water science. Ideas and knowledge. We're going to embed the Headwaters lecture most of the time into the WRS seminar series. So if you're a regular attendee at this, you'll see a couple of these every semester or so. Some of them, if you went to the symposium in January, the first one was embedded there. We may do that again too. So you'll see announcements for these going forward. I'd like to thank Tracy Fallon, who is out right now doing some more coordination as it happens, but who put a lot of effort into coordinating this, along with Dave Mulla and John Niebuhr, who are on the WRS seminar committee and help coordinate all this. So with that, I am really excited to introduce, speaking of internationally known researchers, really excited to introduce our speaker today, Hans Paerl. Who's a distinguished professor of marine and environmental sciences at the University of North Carolina at Chapel Hill. He's stationed at UNC's Institute for Marine Sciences in Morehead City, which is on the North Carolina coast. And among other activities there, he leads the Neuse River Estuary Modeling and Monitoring Program. Hans has earned a number of prestigious professional distinctions, including the G. Evelyn Hutchinson Award from the Association for the Sciences of Limnology and Oceanography. And I think one of the most recent ones was he was named a Fellow of the American Geophysical Union. For other work, but also his work on cyanobacteria and cyanobacterial blooms, he's very well known. He's contributed to a new understanding of the way that eutrophication impacts these blooms. And specifically, there was, for a long time, I think he's going to talk some about this today, a long time there was a paradigm of the understanding that these systems were mainly phosphorus limited, but he led the way along with other people to show that, in fact, they're, in a lot of cases, they're nitrogen limited or co-limited by both N and P. He's also done a lot of work, which he's going to talk more about today, on the effects of climate change on cyanobacteria and how they respond to that. So I had a chance to have a couple of discussions with Hans, both at breakfast this morning and at lunch with students, and these are just fascinating organisms, right? And some of you who study them would know about this, but they're much, much older evolutionarily than humans, 2 billion years or something like that. So humans are just sort of a blip on their radar that have come along. And because they've been around for so long, they're very well adapted to dealing with changes in environmental conditions and, in fact, taking advantage of those changes. So probably in the way they look at the world, human beings and the way we're modifying the environment and loading up aquatic systems with nutrients and the effects we're having on climate change is one of the better things that happened to them in a while. They're doing pretty well. So with that, welcome, Hans. Thank you. Thank you for being here.
Thanks. You sort of gave my whole lecture away already, but you know. So thank you for coming on a Friday afternoon. Hopefully it'll only be a mild snow event. I got to fly out of here tomorrow, back to North Carolina. So my talk's going to be sort of in two parts. First part will deal a little bit reviewing the nutrient issues, the nutrient limitation issues that we're dealing with cyanobacterial blooms. And then I'm going to devote most of the talk to the issue of climate change and how cyanobacteria are taking advantage of that and sort of linking that to their long evolutionary history. And everybody knows this picture, right? I don't need to, maybe I should give a T-shirt maybe to somebody who, what lake? Okay, I don't have enough T-shirts for that answer, so. All right, so just a very quick review here. I think most of you are very familiar with the issues. We know that cyanobacteria are proliferating in lakes, and you know, everybody in Minnesota knows that, but also in many other places. And that, of course, is tied in with human growth, urban agricultural and industrial expansion, which brings with it nutrients, both nitrogen and phosphorus inputs. And, but also hydrology is really important in all this, particularly water use and hydrologic modification. And I'll talk a little bit about that, things like reservoir construction, diversions, things like that. And that's sort of tied in with climate change too, warming and more extreme events. And one thing we're really pretty sure of with cyanobacteria is that these blooms are intensifying and spreading. I think actually we can be more certain about cyanobacterial blooms than marine red tides, which often are far less predictable, and they're really more related to physical changes in the structure of the ocean. So, and you know, contrary to the dinoflagellates that the marine people like to talk about and brag about, we can actually write our names in a cyanobacterial bloom, see? So they actually are more useful than dino blooms. The other thing that's important is that these blooms are not necessarily limited to freshwater systems, and that's something that I think we really need to hone in on because they are spreading in estuaries, for example, and even in coastal waters and seas. And a lot of these genera can tolerate salt. So I think that was another kind of paradigm that was out there for a long time, is that you don't need to worry about them getting into the marine environment. Well, I got news for you. They are out there, and also benthic forms. I'm going to talk about planktonic cyanos mostly, but there's lots of benthic cyanos that are causing problems too, and most of these pictures are actually from the marine environment, from estuaries, including reefs. But I'm going to focus on the planktonic cyanobacteria and the three major groups that we used to call them Annie, Fanny, and Mike. Joe Shapiro, I think, invented that term here at Minnesota a long time ago. So the upper group here are the coccoid cyanobacteria, and they grow in either solitary forms like Synechococcus, for example, or colonies like Microcystis, and I think everybody knows about Microcystis because it's one of the real aggressive expanders. Most of these, almost all of them in freshwater, require external nitrogen. They don't fix nitrogen, and we'll get back to the nitrogen fixation issue in a while. And then the second group are the non-Heterocystis filamentous guys. So they're stacks of cells, filaments, but they're undifferentiated. Some of them fix nitrogen, most of them don't. Actually, most of the ones that are causing problems, like Planktothrix, for example, don't fix nitrogen. So they also require external nitrogen. And then the third group are the Heterocystis guys. Anabina, which has now been renamed Dolichospermum. The taxonomists have great fun with confusing ecologists, but you know, I'll just stay with Anabina for the time being. And Cylindrospermopsis, which is a very aggressive invader of many systems, and you guys need to really look for it because it's moving north. So they can fix nitrogen, and they have those special cells called heterocysts in which nitrogen fixation occurs. Now, what do we need to worry about or be concerned about in terms of how these blooms are regulated or controlled? And obviously nutrients play a big role, and so here's the nitrogen and phosphorus part of the story, coming from different sources. And you know, we're all familiar with surface flows and surface inputs, but we also have groundwater issues in some places with nutrients coming in, and atmospheric deposition, particularly of nitrogen. So there's a lot of out of sight, out of mind nutrient sources that we need to consider in all this. The other factors that are important are physics, And by physics, I'm talking about hydrology, the freshwater flow and throughput in aquatic systems. That determines the residence time of a system, and residence time is critical. It's a critical part of understanding blooms and how they're controlled. In part because cyanobacteria, even though these blooms look dramatic, the cyanobacteria actually are pretty slow growers relative to other phytoplankton groups like diatoms, greens, even dinoflagellates. And these blooms are pretty dramatic, but the cells that are involved in the bloom formation actually don't grow very fast. So residence time is important because you can overcome blooms sometimes by increasing the flushing rate, for example. And this is a popular technique in reservoir management in places. The other thing is the wind and mixing. The cyanos, a lot of the bloom formers are buoyant. They like to dominate the surface waters where they can intercept the light and then shade the underlying phytoplankton and benthic plants. And lastly, temperature, of course, is really important in terms of regulating growth rates and stratification of the water, stability of the water. So the physics are really important. In fact, in many systems, they're almost as important as nutrients in terms of controls. Now, there are also other issues with food webs and grazing and regeneration of nutrients coming from that. I'm not going to cover that, but that can be an important aspect of the bloom formation as well. And so we're going to stick, with the chemistry and the physics here mostly. And I'm going to start with sort of the poster child of excessive eutrophication in cyanobacterial blooms, and this is Lake Tahoe. And in Mandarin, Tahoe means big lake, so I'm just going to use Tahoe, okay? Tahoe. Hu is lake in Chinese, so when you see all these things with hu on them, it's something lake, something lake. And Tahoe is the third largest lake in China. It's located in the Yangtze River Delta, one of the most populated and fast-growing regions of China. It is a lake that has essentially undergone a state change in the 70s and 80s and earlier. It was largely a diatom-dominated lake. And then the cities around the lake expanded greatly with the Chinese policy of moving people into cities. So these cities have grown from less than 1 million, here this is Wuxi and Suzhou, to over 6 million each now. So these cities are discharging untreated wastewater, or have been. It's changing now. So about half the nutrient loading into this lake comes from rapidly expanding cities. The other half is non-point source from a variety of agricultural practices, row crop operations, rice, tea. Actually, tea uses a lot of fertilizer, and I used to think that tea was the best thing. It's still really good, but it takes a lot of fertilizer to grow it. And animal operations, okay, expanding greatly in China as well. So these blooms, and the lake is only 2 meters deep. That's the other thing I need to emphasize to you, because that's important later on. So when you're running around on the lake and you hit one of these blooms, it's sort of like hitting a patch of whipped cream. Your boat slows down, and you can actually physically feel the effect of the bloom on the boat. These blooms can be 10 centimeters or 20 centimeters thick on the lake. And they're out there for about 9 months out of the year. And the amount of time that the blooms are out there now is expanding. And this is part of the global warming issue that I'll talk about a bit later. Okay, so in 2007, Tahoe, they had a very warm early spring. And the bloom got started very early in that year. And it got so severe, it basically covered the entire lake. This is Microcystis, by the way, the dominant bloom former in here. Same organism as out in western Lake Erie. And in 2007, it overwhelmed the drinking water plants for these two cities, Wuxi and Suzhou. And so folks woke up one morning with green jello coming out of their faucets. They immediately knew there was a problem. Nobody drank the water. The central government had to bring in water supplies for about 3 weeks or so while they were getting the water plants back in some functional shape. Sort of like a macro version of Toledo, taking place here in Tahoe. And, you know, the Chinese are really big on engineering and trying mechanical and all sorts of ways of getting rid of the blooms. Here they're picking it up in tanker trucks and driving it off. I'm not sure where it's going, but it's one of the strategies. And the other thing is I just want to point out that politics in China are very similar to politics in the US. Here are the politicians coming down from Beijing showing everybody you can drink the water. It's okay to drink the water. I'm pretty sure they were drinking bottled water there in that picture. But, you know, it's very similar to our politicians, right? They'll go have a fish fry or something or drink water. Anyway, big issue. This led to Tahoe becoming very prominent in terms of the pollution issues China's facing. And it's a revered lake. It's a very important lake in China. So China's put a lot of resources into trying to deal with this situation. Okay, so in a nutshell, here's the nutrient problem in Tahoe. Too much of everything, and sort of a seasonal aspect of too much of everything. So if we look at the T and the TP ratio in the lake, it's very high in the spring. That's because of all that nitrogen coming in from farmlands, basically, coming in after fertilization. So T/N to TP is very high, and then it drops down below the Redfield ratio. This is the Redfield ratio, 16:1 N to P. And then here's 3 years in a row. So you can see this is a very repeated pattern. Very high N to P in the spring, low N to P in the summer, and it repeats itself. And from that, we developed a hypothesis that maybe this lake is actually dual nutrient limited, so that, you know, in the spring it's probably phosphorus limited. Seems like, you know, that would be obvious from the ratios. But in the summertime, it actually drops down below that Redfield ratio, and so there may be nitrogen limitation. So we tested that and have done a lot of experiments since then with all sorts of bioassays ranging from cubitaners to 1,000 liter and greater mesocosms now. And this is the picture that we see when we look at chlorophyll A response to nutrients in the embayments that here's one of these big sort of mesocosm things that have been used too. And what you can see is that in the spring, this is the initial lake water chlorophyll concentration, the control, no addition, nitrogen is nitrate, phosphorus is phosphate, and then N and P together. And what you can see, sure enough, in the spring, there's phosphorus-stimulated growth, okay, because of all that nitrogen that's there. But in the summertime, nitrogen starts to become important, and there's no phosphorus limitation in the summertime. And then it repeats itself the following year, and you can see the second year, actually, the nitrogen limitation is even more severe than the first year, and that probably has to do with hydrology and flow of nutrients into the system. So there's some variability in that, but the picture remains the same, basically, in terms of the patterns. But the important thing to gather from this is that if you add N and P together, you get more than N or P alone. Okay, and that's kind of the take-home message from these bioassays. And we get the same results when we scale up from 1 liter to, I don't know, this here was like something like 4,000 liters or something of water in these big mesocosms. And here's my manager slide for people that don't like to look at data. This is 100 mils of water from the bioassay filtered. This is incubation. I think this is a 48-hour incubation out of the bioassay, control, plus N as nitrate, plus P as phosphate, and N and P together. And usually this convinces people without having to show them any data that, you know, there's a dual nutrient limitation issue there. So the question that's come up is, you know, is Erie just so far out there on the spectrum that it should be really looked at as kind of an unusual lake relative to other eutrophying systems? And I've just given you some examples, some of which you're familiar with, like the Great Lakes, Baltic Sea, Florida's St. John's River, another lake in China, Yanxi, very hard to pronounce. It's taken me 10 years to pronounce that one. St. John's River, Lake Okeechobee, you know, just go down the list here. So what I've done is I've gotten together the literature from studies that have been done or reviews that have been done that have actually looked at bioassays of varying sorts and looking at the results. And these papers, and particularly Jim Elser's paper in 2007, which nicely reviews the nutrient limitation in various trophic state lakes. All these lakes that are on here on the map all have shown dual nutrient limitation, or N and P limitation, or N and P stimulation being greater than N alone or P alone. And some of them actually have even shown nitrogen limitation as well. So Tahoe does not, it's not that unusual in terms of what we see in terms of the patterns of nutrient limitation in the system. And then, you know, the other thing is that there have been lakes where a whole lake has been manipulated and nutrients have been added to the entire lake. And many of you are familiar with the experimental lakes area in Canada, but there have been other places in Sweden and Quebec, Northwest Territory. And this is data that was gathered by Bill Lewis and Wayne Wurtzbaugh, and then we published a paper more recently with more lakes. And what you can see is that the picture is pretty similar to what we see with the smaller bioassays. Nitrogen can stimulate by itself, P by itself too, but surprisingly, in many of these lakes, phosphorus on its own does not necessarily stimulate primary production, and that's kind of a surprise. But look at N and P, much bigger than N alone or P alone. So I would argue that, you know, I mean, you can make arguments that these small-scale bioassays don't really mimic the lake because there's no contact with the sediment, and you know, you got the bottle effect and all these other things. But if you look at them on the right time scale, and for these small bioassays, they're like a snapshot. You know, you look at a few days. If you do the whole lake edition, you can look at it for a whole year or a whole season, but they give you the same answer, more or less, in terms of the nutrient limitation question. So, what we've concluded from this is that, you know, here's sort of the result, the overall result from these lake experiments, whole lake fertilization experiments, is that, you know, it takes two to tango to really get the maximum oomph in terms of the eutrophication potential for these lakes. And that was published in a recent paper that we had in Environmental Science and Technology. And if any of you are interested, I'd be glad to send you that email. So the question has come up, well, you know, if nitrogen is limiting, why doesn't nitrogen fixation make up for this limitation? Because we got all these cyanobacteria out there, many genera that can fix nitrogen. And in fact, they do fix nitrogen. But the issue is not as simple as just having enough phosphorus to sustain nitrogen fixation. And so this is the enzymology, essentially, of nitrogen fixation. Here's the enzyme, the dinitrogenase. And here's the phosphorus connection. You know, phosphorus is needed to make the high-energy compound to support this process. But there are other things that come into play in terms of controlling nitrogen fixation, and one of them is energy, the actual source of energy to support the process and also support ATP formation. And of course, with cyanobacteria, it's photosynthesis. But with bacteria, it could be organic matter and other energy-rich compounds. And if you can imagine a highly turbid lake like Tahoe, where the Secchi depth reading can be like only 5 centimeters or something like that, or 10 centimeters, you can see where light limitation might be a problem if you're going to have to fix nitrogen to get your nitrogen quota up, okay? So, and I'll get back to this in a second, but so light can be a big issue in terms of controlling N fixation. Other things could be cofactors like iron availability, for example. And let's not forget about oxygen. And you know, we often ignore oxygen in looking at what's limiting things in lakes. But when we have these big blooms, the oxygen saturation during the day can be 200 or greater % saturation. And in experiments that we did way back in the 70s, that is the last century, we actually looked at lakes in New Zealand and took fresh samples from those lakes that had nitrogen fixers in them, and we exposed those to different oxygen tension. And you could see that even with the heterocystis cyanobacteria, they did not perform as well under under supersaturated oxygen conditions than they did under low oxygen conditions. So even the heterocystis cyanobacteria are somewhat limited by excessive oxygen. It's not a perfect, it appears to not be a perfect protective mechanism, just having those heterocysts. So it's not, the bottom line is it's not just P, but we're talking about energy requirements, micronutrients, and then last but not least, oxygen can be a problem. So we think that nitrogen fixation, the reason it's not fixing or it's not able to match the phosphorus in the stoichiometric sort of ratio is that there's limitations other than just how much phosphorus there's present in the water, okay? And if you look at the two processes by which nitrogen enters the system, N-fixation and denitrification, and we have some lakes here where pretty good measurements have been made. This is in a paper that Thad Scott and I published back in 2010. And we look at it on an annual basis. You can see the flux is always outward. So denitrification is exceeding N-fixation in these lakes. So they're always losing nitrogen on a net basis, okay? And these, this is the main biological loss mechanism and entrance system, or entrance. So how does this all work? And how do you make the argument that nitrogen limitation is kind of a chronic issue in many lakes? So we have our nutrients coming into the system. Black is N, orange is phosphorus. High nutrients stimulate primary production. We know that really well. That production then drives heterotrophic processes like denitrification and nutrient recycling in the system. But it also drives denitrification because that's a heterotrophic process. And we know now from these other studies that in most of these eutrophic lakes, denitrification is greater than N-fixation. So you got this loss going on all the time, even in lakes where nitrogen fixation is going on. And phosphorus, of course, does not have an atmospheric component. It doesn't have a gaseous component. So phosphorus is more or less locked in here. It really is mainly lost by advective processes, vis-a-vis the legacy issue with phosphorus loading in lakes. So these lakes are always nitrogen hungry. They just, and also you can say the coastal systems and coastal waters. So what that means is that what's coming in externally can make up for that nitrogen deficit and maintain the high eutrophic conditions, okay? Everybody follow me there? Because this is really the kind of the crux of the argument here. All right. So the second part of the talk deals with climate issues. And we all know that things are warming up. This is the North Atlantic anomaly. And very nice data from the Netherlands looking at epilimnetic temperatures in Dutch lakes. And you can see that it's gone up for over 100 years. And also in Lake Erie, this is data from George Bullerjean et al. So the evidence is certainly there. And I even have evidence for someone like Donald Trump that climate change is happening. So you mentioned that he might actually like this slide, and I'm kind of worried about that because maybe I don't want to show this anymore after this talk. All right, so why does temperature matter? So if we increase temperature, we're going to enhance stratification. I think everybody who does limnology knows this. You create a greater density gradient in your epilimnion in the system. Okay, so that creates stronger stratification, and under stronger stratification conditions, the buoyant cyanobacteria are going to benefit because they like to be at the surface. Okay, so you're kind of optimizing their ability to dominate those surface waters under strong stratification. And here's data again from the Netherlands on Nieuwe Meer, which means new lake in Dutch, actually. It was probably a lake that was created from a polder, I think, in 300 years ago or something in the Netherlands. But you can see here over time, this is just going from spring into summer. You can see the heating going on, and then you see the strong stratification developing in the epilimnion. And here are the cyanos. They initially respond, but they really respond strongly when you get that strong stratification in the system. And the interesting thing about this story is that this lake is pretty much nutrient saturated. So, we're really looking at physics here driving the process, okay? So that's kind of nice too. It's a big problem having all those nutrients, but you can look at the physics really nicely. And Microcystis benefits under those conditions, and you have these persistent surface blooms. And Jeff Huisman and his group at the University of Amsterdam did some they cordoned off part of the lake and mixed part of the lake and left the other part, they allowed the lake to stratify on its own, and then in the mixing part, they used aerators to mix the lake. And what you can see is that when the mixing's off, when the stratification is allowed to occur, the cyanobacteria dominate in the summertime. Mixing on, the diatoms dominate because they're catching up. They grow faster than cyanobacteria, but you got to give them the conditions by which they can compete, which is under mixed conditions. The cyanos, the diatoms are kind of dumb in that they sink and allow the cyanobacteria to dominate the surface waters when you have that strong stratification. So this shows again, the physical control really strongly, particularly under. Now, another thing that Jeff pointed out to me when we wrote this paper back in 2008 is that the viscosity also is affected by temperature. And when temperature warms up, there's lower viscosity in the water. And that allows buoyant organisms to move up faster, but also it allows organisms that sink to sink faster. Okay, now this hasn't really been explored yet to any great depth in lakes, but it would be another factor that would actually benefit the buoyant cyanobacteria. And then lastly, temperature itself affects the growth rate of the phytoplankton that are competing. And so here we're looking at temperature versus growth, percent maximum growth, diatoms, dinos, chlorophytes, and cyanos, and you can see that things are skewed towards the warmer part of the scale here with cyanobacteria. So we know now from other experiments, lab experiments, for example, that cyanos like it hot. So there's a direct relationship there between warming and their ability to compete at better growth rates at those high temperatures. And this is some interesting field data from Kosten et al., another Dutch group. I'm biased towards the Dutch here, but they've done a lot of work with cyanobacteria. And what they did was they sampled, how many lakes? 143 lakes from the tip of South America all the way up to the Arctic Circle. So they covered this entire latitudinal gradient. And what you can see is that when they plot temperature against biovolume, cyanobacterial biovolume, you see at the warmest temperatures, the cyanos tend to dominate. And if you put nitrogen into the equation, Tn, you get an even better relationship. So here, actually, you can form a relationship between Tn and temperature to see how that cyanos are clearly promoted when the Tn levels are higher and you have higher temperatures. Now, you can plot Tp in this as well, but actually the relationship is not as strong as it is with Tn in this particular relationship. So here's field data basically substantiating some of the lab data and physiological data. So now I'm going to go into some speculation here on the Great Lakes. And we all know that Lake Erie has the biggest problem with cyanos, but there are other places like Green Bay, for example, and appendages from other lakes. But Lake Erie is by far the biggest problematic lake with cyano blooms. But it's also warming up at the same time. And it is the warmest of the Great Lakes in terms of overall temperature. So this is suggestive that there may be some sort of interaction here between temperature as well as nutrient loading in this system as well. And this may be implicated in this resurgence that we're seeing of cyanobacteria in Lake Erie. It's still, I think this is still highly speculative, but it certainly fits. It fits well between the nutrient loading, excess nutrient loading, and warming. And the dominant organism, again, I just want to harp on this a bit, is Microcystis. It does not fix nitrogen. It cannot fix nitrogen. So that's telling us something about nitrogen. Okay? And of course, right now everyone's trying to figure out how to deal with these blooms in Lake Erie. And the obvious thing is to reduce phosphorus, but we've reduced phosphorus quite a bit, although in the Maumee River, where these blooms are occurring, there is elevated phosphorus being discharged, but also elevated nitrogen. So we cannot ignore nitrogen in this equation. And the other thing is that, Erie has had excessive phosphorus loading for a long time, going on, probably even before World War II, but certainly with the advent of fertilizers and wastewater discharge to it. So this legacy may be haunting the ability of Lake Erie to recover if you add more nitrogen on top of the phosphorus that cannot escape the lake. Okay, so this is sounding like a pretty gloomy story, but I think what it's telling us is that we really need to deal with nitrogen probably too. Okay, and since we know that nitrogen fixers, at least from the lakes that have been looked at, cannot match the nitrogen requirements that are needed to balance the sort of stoichiometric ratios, I don't think we need to be too afraid of reducing nitrogen in these systems to try to improve water quality conditions, particularly the cyanobacterial blooms. Lake Biwa, largest lake in Japan, it has gone through warming as well, and you can see that here's the water temperature and the annual or sort of ongoing water temperature that's been monitored there. So, sort of a similar story to Erie. They did phosphorus reductions. It improved somewhat, but now they're sort of back to a more or less constant situation with cyanobacterial blooms. And even though phosphorus has been reduced pretty drastically there, and most of that's point source phosphorus, by the way, so pretty large percentage of the inputs being controlled, they're getting a resurgence of blooms in Lake Biwa as well. And then my favorite lake, where I started my career, Lake Tahoe on the California-Nevada border, one of the most oligotrophic and beautiful lakes in the world, has also undergone temperature increases. And this has been documented by Charlie Goldman's group and others. So even a large lake like Tahoe, which has a huge volume of water, is undergoing warming as well. And the question then, of course, is, we know from Charles's data, from the primary productivity data, he has, this spectacular graph showing the increase in primary production in Tahoe, which has largely been attributed to excessive nutrient loading, and that's probably true. But the question also is, how much does this interact with the nutrient inputs in the lake to actually exacerbate the situation? And of course, the jury's still out on that, but all these things are going on even in an ultra-oligotrophic lake. And then Tahoe, which doesn't have as long of a data set as Tahoe. So I've worked on 3 lakes that all sound the same. Tahoe, and then I was in New Zealand and worked on Taupo, and now I'm working on Tahoe, and I'm looking for another lake. Minnesota's got like 10,000 lakes, right? So somebody come up with a... Okay, so in Tahoe, there's good data on air temperature, okay, but not so long a data record on water temperature. So here's the air temperature. This is the mean air temperature between between the end of April and the beginning of June, when the blooms first start in Tahoe. So and this is at Shanghai, which is about, 80 kilometers or so from Tahoe. And you can see that, things were pretty constant, and then we've gotten into this warming trend in the 90s. And here's the surface water temperature data in Tahoe, and it seems to track the air temperature, which makes sense. And here's chlorophyll A. Now, of course it's gone up, but the loading in Tahoe, the nutrient loading in Tahoe, has more or less stabilized in the lake. And yet the chlorophyll is still going up in the surface water. So we think that maybe there's also a relationship here between temperature and the eutrophication potential, which would be really problematic because we're trying very aggressively to control these blooms in Tahoe. But what that means is that the bar has been raised, more. So we probably need to reduce nutrients even more to meet the high growth potentials at the higher temperatures. So this is not a good news story, but it's something we need to deal with climate change occurring. And then lastly, hydrology has changed a lot. You know, storms have gotten more extreme, and droughts have gotten more extreme, and this is really more of a North Carolina story here, but here we have 3 hurricanes in the Atlantic. If I don't ever see another hurricane, I won't miss them. And of course, they bring with them tremendous slugs of water, high nutrients, but we also have more extensive droughts. So, you know, everything's getting more extreme, more extensive high flushing conditions and more extensive droughts. And if we look in one of our estuaries that we have a long-term data set on, you mentioned the ModMod program. This is data from the ModMod program. We look at the phytoplankton groups based on diagnostic pigments. And the fucosanthin encodes for diatoms, and zeaxanthin here encodes for cyanobacteria. And then here's the discharge. This is the freshwater discharge coming into the estuary. And what I want you to do is just follow the arrows. So when we have high discharge events, the diatoms come on strong because they can grow fast. And they like high nutrients, even when the residence time is short. And then when we have our drought periods, the cyanobacteria dominate. So there's also this hydrologic forcing going on at the same time. So we've got temperature effects affecting, you know, lake stratification, buoyancy, growth rates, and now we also have hydrologic issues to deal with in terms of what's controlling the competition between these groups. But it's pretty convincing data, and it goes on and on and on. And I could plug more years in there, and you get the same kind of thing. So diatoms like it cool and fast. Cyanos like it hot and slow. I don't know, that sounds kind of not the way it's supposed to be, right? Okay. All right. And then if we go to the New River estuary in North Carolina, which is just down, it's down the coast a little bit from Morehead City. It's a semi-lagoonal estuary, so it has pretty long residence time. And we had the good or bad fortune of getting a hurricane or a tropical storm coming across, but we had in situ vertical profiling there, very similar to some of the units that you guys are using at the lab down the road here. And I'm just going to explain to you the data here. This is temperature, salinity, oxygen, DO, chlorophyll, and turbidity. And what you can see when the hurricane or when the storm passed, everything got mixed. So everything's uniform more or less in the system. Here's the chlorophyll. And here's the big turbidity slug from the resuspension of nutrients or sediments into the water column. And what I want you to pay attention to is the chlorophyll levels, which are actually higher after the storm passed, okay, in the system. And that's because the resuspension of nutrients that went on with the hurricane is releasing nutrients into the water column. And the phytoplankton are doing really well. In fact, they do so well that we get blooms in the system, including cyanobacterial blooms. So here we have a storm event that's bringing in more nutrients, in this case through resuspension, although there's probably some coming in from freshwater discharge to the system. You know, we get different hurricanes. We get like windy and dry hurricanes, and then we get wet hurricanes that get stalled and they rain a lot over the coast. This was a dry, fast hurricane, okay, over the system. And then if we go, and then another example of physical drivers in this system. This is dinoflagellate, carlidinium. Some of you might know it. It's a fish killing. It produces a toxin that plugs up the gills of fish and that causes them to suffocate. And what you can, this is after Tropical Storm Ernesto, which visited us in 2006. Here's a side view of the Neuse estuary. So this is the headwaters, the freshwater. This is the entrance to Pamlico Sound, and this is salinity. And you can see the salinity stratification developing with that fresh water running over the salt water. Here's the gradient, the salinity gradient in the system. And right where the gradient is really strong and you're getting that stratification, a bloom develops. So it isn't just cyanos. There are other characters out there that can take advantage of these events as well. So, you know, we've got this runoff associated with high nutrients, sets up strong salinity stratification, followed by favorable light and temperature conditions, and they created the ideal conditions for a bloom. And then this is dissolved oxygen, by the way, that's produced by the bloom. This is chlorophyll A. So this was in a pretty confined part of the estuary. These lines here are sampling profiles for the different stations in the system, and you can see this is a very confined event. And if you don't have good monitoring in place, you could miss this whole thing. And within a week or so after this happened, this is the cells here. This is the toxin production, which was analyzed by Al Place up in Maryland. About a week after this happened, we had fish kills on both sides of the estuary, right where that location is, right over in here. So this is real good data to explain not only how these blooms form, but also why we're getting fish kills in this system. And we know that low oxygen kills fish, but also we do get some of these bad players in there that can lead to that. And then lastly, Tahoe. And Tahoe gets hit by typhoons because it's more or less at the same latitude as North Carolina. It gets the typhoons that come in from the Pacific. And I had the good fortune here of having years where we had a typhoon, and they mostly come in August. They're more regular than they are in North Carolina. So here's 1 year with the typhoon, no typhoon here, typhoon there. And this is just chlorophyll A remotely sensed over the system. So you can see that probably due to nutrient resuspension, but also inputs of nutrients from the storm event, can have a big event on blooms and restarting the blooms as long as the temperature is still high enough to develop the blooms. These are mostly Microcystis blooms. So just to go over what I've talked about, and hopefully I've left some time for questions. Nutrients are important, both N and P, we think. Flow and hydrology is really important. Wind speed and mixing are important. And warming, of course, in this whole scenario. So we have this sort of interactive set of events that's going on here that's controlling these bloom phenomena in either lakes or estuaries. Now, what's the knob we can tweak in all this? The one knob we can tweak is the nutrient knob. We can't change the climate, at least not in the short term. So my argument would be is that, we need to take a longer and harder look at nutrient thresholds in these systems, particularly if they're changing and the thresholds are changing to allow the cyanos to become more dominant just by physics in the system. So the take-home messages, got to deal with both N and P in most cases. There are probably some cases where you can get away with only P or N reductions. The thresholds are probably system-specific, the nutrient bloom thresholds, but it's been our experience and experienced by others that we're looking at pretty significant reductions of both nutrients in many of these systems, probably greater than 30%. And that should not come as a big surprise, because if you look at historic loads, trends in loads, they've increased by at least those amounts in the system. And of course, we may need to be even more aggressive in a warmer world because we know blooms like it hot, and episodic events favor these blooms. And lastly, the nutrient restrictions need to be year-round because most of these lakes and even estuaries have pretty long residence time, at least a few months or so. So the idea of only controlling nitrogen in the spring and controlling phosphorus in the summer, and if we include the legacy issue of these nutrients and systems, I think we're looking at year-round controls of nutrient input. So this question's been asked by managers. Well, can we get away with only nitrogen reductions in the spring and P in the summer? And my argument, based on legacy and internal cycling, is that that's probably not likely to be successful in the bloom control. So I just wanted to thank some of my co-workers, particularly my Chinese colleagues, who have done a lot of great work at Tahoe, but other lakes in China. This is Tim Otten, who was my PhD student looking at toxins associated with Microcystis blooms. And just wanted to acknowledge the support by NSF and EPA and internal support in China by Chinese Academy of Sciences and the Ministry of Science and Technology. which actually supports our activities when we're in China. So this really saves a lot in terms of, getting grants to do the work, because we don't have to pay for stuff once we're over there. And lastly, don't forget about nitrogen. I just wanted to give you that take-home message too. So going to end there. Thank you very much, and I'm more than glad to answer any questions and or respond to statements or whatever. Thank you.
Are you asking a question? Yeah. Because we have a
Well,
microphone.
we will bring you the mic so you're properly recorded.
Thank you for your excellent presentation. I've also been asked to go over and work with the Chinese government on Lake Tahoe or Tahoe. And I have a couple of questions for you. First of all, there's a lot of air pollution, and the sunlight is really limited. Does that have any role to play in the nitrogen limitations that exist?
Yeah, we don't think so. I mean, the actual surface irradiance is pretty high in Tahoe, even with the air pollution. I mean, we get like 1,500 micro-Einsteins in the surface layer, in the surface waters. And the saturation, if you look at the actual light saturation, it falls quite a bit below that in terms of the optimal growth for the cyanos. I think a bigger issue is the turbidity of the systems in terms of light limitation.
Right.
And that's true not only for Tahoe, but if you go to other lakes in China. Have you been? Yeah, you've
Yeah, I've
been
been
to
to
other places?
Poyanghu and Tahoe.
Yeah, I mean.
Right. Both Poyanghu has terrible sediment
Yeah, and
problems.
Chaohu,
Yeah.
Lake Chao.
So the second question is that, from what I understand, they've set a limit for nitrogen concentrations in the lake of 1 milligram per liter. That seems like it's extremely difficult to attain. I just wonder why such a low limit. I mean, here in Minnesota, for example, our water quality limits for eutrophication and nitrogen related would be much, much higher than that. Is it because the lake is so shallow, or is there some other reason?
I'm not sure what that's based on, to be honest. We're finding, so we've done dilution bioassays there too. And the idea of the dilution bioassays is to dilute the water to see what the threshold is. Where, where does, if you dilute the water with major ion solution that doesn't have nutrients in it, you'll get to a point where, growth is limited. And then you add back N and P to see which one is actually the limiting nutrient at that point. And when we do those reductions, or when we do those, it looks like we need to reduce phosphorus concentrations in the lake by somewhere around like 40%, and nitrogen even more, 50 to 60% or so. So looking at that relative to what we see in the spring with the maximum nitrogen input to the lake, that can be like 2 to 3 milligrams, and most of that's nitrate. So I'm not sure if 1 milligram is attainable there. So I would question that as well. But, I think it's really the only strategy we can use for the lake, because other things have been done to the lake to try to alleviate the blooms. The engineers got in the room first with the party members after that those disastrous blooms, and they convinced there was tremendous pressure to do something about the eutrophication of the lake. And the engineers, they always have an answer, We can build a dam, we can flush this, we can do that. So they basically convinced the Chinese government to connect the lake to the Yangtze River and flush the lake with Yangtze River water to physically flush the bloom out of the lake. This lake is like 23, it's big. It's 2,300 square kilometers. And the best they could do with reducing the residence time was to go from about a year to 220 days or something like that. And, if any ecologist had been in the room when that decision was made, they would have spoken up and said, you cannot, you know, the algae can exceed that in terms of their growth rate. So, and then the other problem with diverting water from anywhere in China into the lake, let alone the Yangtze, is you're putting very high nutrient water into the lake trying to flush it. So in a way, the diversion project in the lake, which by the way costs over $2 billion US, has actually raised the bar because it's increased the load by 15% to the lake itself. The other strategy that's been used, and it's still going on, is dredging in some of the embayments of the lake, with the idea of getting the nutrients out of the sediments. The problem with that is, again, it's a big lake. And then what do you do with the dredge spoils, in a watershed that is huge? So what they do with the dredge spoils is just put it up on a hillside above the lake. So all you've done is put another loop there in terms of the nutrient cycle. And that has cost about another $2 billion US. So, what they should have done was to start advanced wastewater treatment from the cities, which could have reduced the load by almost half to the lake, and then put in these things that we're using, for non-point source control, like riparian buffers, wetlands around the lakes, around the lake, and stricter regulations on fertilizer application in the watershed. Some of that's going on now in the Tahoe Basin. So, I'm somewhat hopeful that they are going to make advances, but it's going to be a slow process in the lake. And, the storage of pee in the lake is a huge problem, too.
Hey, hi.
Hi.
That was awesome, first of all. And second of all,
Thank you. I need you in the audience all the time.
oh, well, I'm happy to be like your fan club. So I actually have a question that's not related to nitrogen, but early on in your presentation, you showed results from a nutrient limitation study that you did. And I noticed that there were quite a few phosphorus inhibition scenarios.
Yeah, well, we get that sometimes with bioassays. Not often, but we do get it, and I've actually seen it in some of our estuarine systems too. Now, the estuaries they're never phosphorus limited because there's so much phosphorus in coastal systems. But in some of the Florida lakes that we worked in, we do get sometimes lower than control chlorophylls with P additions. And the only explanation I have for that is that it's precipitating something else that may be limiting, like iron, for example, in the availability. Other than that, I have no explanation.
That is something that I'm going to look into, because this has been bugging me for about 7 years.
Yeah, well, it's been actually, if you go to Jim Elser's paper, I think he also found some of that.
That's awesome. Thank you.
Yeah, that could be used as an argument for putting more phosphorus into systems. Any more?
Well, Hans, thank you so much for your presentation and sharing your thoughts with us.
Yeah, well, I'm delighted to do that. And my roots are in limnology, so it's great coming back to a place where limnology almost started in the U.S. So, I'm a great fan of some of your faculty, your earlier faculty, like Joe Shapiro, for example, who I think was a absolutely wonderful scientist, and he really got me interested in a lot of the blue-green stuff that I did in New Zealand.
Oh, cool. Well, it's been great having you, Minnesota. Let's thank him one more time. And if you can stay and join us for a reception, there's refreshments just outside the door in the hallway, so you can interact.